All right, so we are in chapter 6.1 and we are going to talk about viruses. So, kind of our understanding of viruses goes back kind of all the way back to the 17th century. So, in the 17th century, people were doing something called variolation. And we'll talk more about variolation when we get to actually talking about vaccines and things like that. But it's similar to vaccination, where people were actually taking things from people that were sick. So they were taking things out of their noses and taking sputum and things like from saliva from their mouths, and they were putting it into other people's bodies. So they would actually blow these things into other people's noses, and then they would get sick. And then, since they didn't get lots and lots of the virus, then they would actually become immune to it, which we know now and in which we call vaccination. And that actually started in the 18th century. So in the 18th century, they actually started doing what was called vaccination. And we'll talk more about vaccination when we get to the chapter on vaccination. But this is kind of showing that even in the 17th century and the 18th century, that people were kind of had somewhat of an understanding that there was something very, very small that could be moved around from person to person and caused illness or could prevent worse illness. Now, it wasn't until 1892 when a gentleman named Dmitry Ivanovsky, so Dmitry Ivanovsky, he's actually the person that is kind of named for starting virology. He's kind of the person that found the first virus, kind of recognized the first virus, and then started what we now call the study of viruses or virology. And this is because what he was doing was he was studying the tobacco mosaic disease, or TMD. So tobacco mosaic disease. And he was studying the tobacco mosaic disease, and he actually used different size filters. And when he was filtering out material from the tobacco or from plants that had the tobacco mosaic disease, he used a filter that they knew would catch all of these bacteria, because bacteriology had been going on for quite some time. They're using a filter that should catch all the bacteria, and then what he showed was that something was still infecting these plants. And so then other scientists were continuing the work on this. They looked even further and they concluded that there was something that was infecting them, but it wasn't as big as the bacterium. They thought maybe it was like a specific molecule or a kind of toxin. And so then this is really what started the virus, because virus actually comes from poison in Latin. And that's because the scientists they didn't know what was going on, but they thought maybe it was a molecule that was a specific type of poison that was causing this problem, because when they were using these very, very fine filters, whatever this was still getting through and infecting the next plants. So Dmitry Ivanovsky kind of is credited for discovering viruses. Even though he's not the one that directly saw one, because that didn't happen until the 1930s. And the reason that didn't happen until the 1930s is because it was then that we had microscopes that would allow us to see something this small. So this is when we had our electron microscopes, because they are so small that we couldn't see them with our regular bright field microscopes or any of the other stronger microscopes at the time. We had to wait until we actually had developed the electron microscopes, and then we could see something the size of a virus and kind of confirm all of the work that had been done from 1892 all the way up until the 1930s, when the electron microscope came into existence. So what are viruses then? So if they are these really small things that aren't quite like bacteria, what are they? So first of all, viruses, as we know, are acellular. All right, so acellular, A meaning without, and cellular, a cell. So without a cell. And that's why it's not technically a living thing, because recall, all living things, the smallest living thing is a cell. So a virus is not considered a living thing. And that's also because they are obligate intracellular parasites. And what this means is that they are obligated, that's where the obligate, so they are obligated to be in, so intracellular, in a cell. So they're obligated to be in a cell to live, meaning that they have to be within a cell in order to live. They cannot live outside of a cell. And so the reason for this is because they don't come with all of their own machinery to make their own copies of themselves. They come with very, very minimal pieces. And those very minimal pieces, what they do is they then hijack an actual cell, a living cell, and then force that cell to make the copies of the virus. So they utilize all of the machinery inside a cell. They can make dozens or thousands of copies of themselves, and then they burst out of that cell, and all of those copies that now exist can't live or can't make copies of themselves without being in a new cell. So then they all go out and they all find a new cell and then do the same thing. They get inside of the cell, they hijack the machinery of the cell to make copies of itself, and then once it's all filled up with copies of itself, then that cell bursts and they continue on. So obligate intracellular parasites, meaning that they must infect a host to survive and reproduce. So they cannot survive or live very long outside of a cell, and they absolutely cannot reproduce without being in a cell. That's required. And so when it goes through this process, when a cell goes through the process of infecting a cell in order to hijack the machinery and make copies of itself, when it makes those new copies and they're all within the cell, those are called virions. So virions are the newly made virus particles. So the newly made virus particles, after it has hijacked the machinery of the cell, makes lots of copies of itself, and then when they burst out and they're starting to move within, for example, the bloodstream in an animal, then they are viruses. And then those viruses infect the next cell, and then the new copies that are made are called virions. So overall, viruses, not only are they acellular, so they are infectious, acellular pathogens. So they always infect the host, right? So they get inside of the cell and they make copies of themselves. They're acellular, we already mentioned. We already mentioned that they are obligate intracellular pathogens, or obligate intracellular parasites. And with this obligate intracellular parasite characteristic, it's with a specific host and even sometimes a specific cell type. So with a specific host and or cell type. Meaning that viruses, there are viruses that only infect plants, so they wouldn't be a virus that can infect any other animal. There are viruses that only infect a certain type of animal, so say they only infect birds, or they only infect pigs, or let's say they only infect humans. So they are specific to hosts. However, there are some that can go to different hosts. So it's not necessarily one specific host. Maybe it can infect one or two different species. And so if we say that a virus can infect one or two different species, that's its host range. So its host range is the number of species, or kind of generally the number of species that can be infected, or number of species of virus can infect. And I'll say species / cell types. For example, the avian flu, right? So that was infecting birds, but it also infected humans, and it could be passed along. And there are different types of viruses that can do that. And we'll talk more about that in just a minute. So we have different hosts And we also have different cell types. So, for example, some viruses are only going to infect respiratory cells, or some viruses are only going to infect eye cells, or so very specific cell types. And that really depends on the specific virus it is and how that virus works. Also, viruses, let's see, another characteristic is that they have RNA or DNA, but they never have both. So RNA or DNA, never both. Right, so when we talk about a eukaryotic organism, think that we have DNA in our nucleus. We also have RNA that's being made from the DNA. A virus only has RNA or DNA. It doesn't have both of those things at the same time. So it can come in with DNA or RNA. Its structure, so when we talk about the structure of a virus, the general structure is that we have the genome, so that RNA or that DNA, and that is surrounded by what's called a protein capsid, or it can have a phospholipid membrane. And so it can have a protein capsid around it, and then it can also have this phospholipid membrane around it that has spikes in it. So it has a genome with, rather than saying with, we can say surrounded by a protein capsid. So we have our genome, whether it's RNA or DNA, surrounded by this protein capsid, and then outside of this protein capsid, sometimes we have this phospholipid membrane that has these glycoproteins on it. Sometimes a phospholipid membrane with glycoproteins. And these glycoproteins are called spikes. And we'll talk more about that in the next section. So we can have just the capsid around the genome, or we can have the capsid and this phospholipid membrane. And then lastly, it does not have the genes that are necessary for reproduction. And we already mentioned that a little bit, but we'll put that here. So it lacks genes for, we'll say, complete reproduction. So again, the virus has to go into an actual cell, obligate intracellular pathogen, and it has to get into the cell and hijack that machinery so it can make RNA, it can make copies of itself, it can make its DNA if it has DNA, and then kind of piece itself together and then burst out from the cell. All right, so let's talk about how we can transmit viruses. So how a virus works. And we already mentioned the host range. So remember that the host range is when we talk about how many different kind of species or how many different types of cells that a virus can infect. And sometimes, most times, it's just one species. But sometimes it can be one or two species or different types of cells. So we have the host range. And a lot of times the virus is going to be named based on that type of host. So it'll be a plant virus or it can be an animal virus. Now there is one that you should know that's specific, and it is called a bacteriophage. And a bacteriophage is a virus that infects a bacteria. So a virus that infects a bacteria. I'm going to erase this E here to make it a little bit more clear. There we go. So a bacteriophage is a virus that's going to infect a bacteria. So if you can imagine how small a bacteriophage must be, because it has to be small enough to infect a bacteria, get into the bacteria, make lots of copies of itself, and then get out. So a bacteriophage is an important distinction. You should know a bacteriophage because as we move through different chapters here, that's going to become important because we actually use bacteriophages to further our knowledge about viruses, to further our knowledge about bacteria, also to build different types of bacteria that include different things like certain antibacterial properties, so we can move those genes back and forth between different bacteria using bacteriophages. So bacteriophages are very, very helpful in the laboratory setting and in the medical field. So bacteriophages are those that infect bacteria. And then we can talk about how we transmit different viruses. And so there are three general ways that we transmit viruses. So the first one is through direct contact. So direct contact, I think you can figure out what that means. So it's directly contacting the person that it's passing to. So this would be, for example, if somebody is infected with a virus and they sneeze on another person. Or even if they don't just sneeze directly on the other person, but if they sneeze and then the person walks through that sneeze cloud, for example, that's direct contact because it's going directly from one host to the next host. It can also be, so, coughing, sneezing, sharing blood in any way on accident or purpose, touching somebody that has some of the virus particles or some virus on that body part, hand or whatever. Say they just sneezed and, caught it with their hand, and then they go to shake hands with somebody shortly afterwards. So that's direct transmission or direct contact. The second one is indirect contact, and indirect contact with fomites. So what is a fomite? A fomite is an object that can transmit. So a fomite is an object that can transmit, we'll say, a microorganism. Because a fomite can include also transmitting bacteria. So in this case, what we're talking about for fomites, we're talking about viruses, but this would be an example where, say, that person did sneeze and they kind of caught some of that sneeze with their hand, and then they go to leave the room and then they put that hand on the doorknob to open up the door and to walk out. Well, they likely have then just transmitted some of those virus particles onto the handle of the door, and then another person can grab that handle and then get the viruses in their body and then become sick. So the fomite in that situation would be the door handle. Fomites also can include things like children's toys or blankets or stuffed animals. They can include silverware. They can include cups. They can include counters or, tables. So anything that can transmit something that is not the actual host. So, sneezing or coughing or wiping something, somehow getting that virus from the host onto that object, and then another host touching that object and then becoming infected. And then the third way is through a vector. And when we talk about vectors, we have different types of vectors. A vector, in general, is an animal that's going to transmit a pathogen from one host to another. So an animal that transmits a pathogen from one host to another. So in this case, it's kind of similar to a fomite, but in this case, it's an animal that's transmitting it. So there are some common vectors that you can probably think of. So common vectors are going to be things like arthropods. So things like ticks, mosquitoes, flies. So these things are all arthropods that are animals that are going to be able to transmit a pathogen from one host to another. So carry it from one host to the next host. And there are actually two ways that they can do this. So one way is if we have a mechanical vector. So a mechanical vector means that they are carrying that pathogen on the outside of their body. Carry pathogen on outside of the body. And so what this means, for example, is if you are at a gathering and you have a fly that lands on someone's food, and that food happens to say their fork, right? So somebody that's sick is using a fork, they're putting it in their mouth while they're eating their food. They set the fork down on their plate, and then a fly lands on that fork. That fly now has that virus or some virus particles on the feet of the fly. And then that fly flies over to the next person's food and lands on their food to eat some of their food up. And then they have now just transmitted that virus from one host to the other host. But it didn't include actually being inside of that animal. That fly didn't, ingest it. That fly wasn't infected in any way. It just had it on its body. So we can see this in different ways. It being on the outside of something, and then the human, comes in contact with it and then becomes infected because of it just mechanically getting from one place to another. The second way is if it's a biological vector. And a biological vector then is when it's carrying the pathogen inside its body and then transfers it through biting. So inside the body and transfers through biting. So in this case, this is a common one that we think of with mosquitoes. So for example, mosquitoes and Lyme disease or dengue or something. So where the mosquito lands on the first person, the host pulls some of their blood out, and pulling their blood out pulls out the virus as well, gets it inside of the mosquito continues flying on, lands on the next person, puts their proboscis in that person, goes to pull out more blood, and what it's doing is it's exchanging the blood that's inside the mosquito from the first person, the host, and then it's exchanging that with the second person, the new host, and so that virus is transmitted in that way. We see the same thing happening with ticks, right? So a tick lands on one animal, it takes some of the blood of that animal, including any viruses that it contains, and then that tick ends up landing on another animal at some point, and then in that process of taking the blood from the second animal, it transmits that virus. So that's a biological vector because it carries the pathogen inside of its body, and then once it bites the person, it's exchanging that genetic material or exchanging those fluids, for example, and then is getting that virus in the next person. So mechanical is just mechanically moving it from one place to another place, on the outside of the animal. Biological means it's actually getting inside of that animal biologically, living-wise, and then is moved to the next person by biting in some way. So lastly, in this one, another definition that we need to mention is zoonoses. And this is what I had mentioned previously, so now I want to put a term with that. And a zoonoses is a virus that's transmitted from an animal to a human and causes a disease in that human. So in this case, what we're saying is we had talked about the host range, and now if the host range is going to go outside of its species between a human and an animal, then we call this a zoonoses. And so, for example, the avian flu that I gave, so it originated in birds, it started out in birds, but then it was transmitted to humans and it caused disease in humans. And so that is a zoonose. That's a virus that is transmitted from animal to human and causes disease in the human. Okay, so zoonosis is when it's going to go across species there. And then there's actually reverse zoonoses. So that's when there's the infection by an animal, by that particular virus, is in the human, and then it can go to something else. So sorry, let me say that more clearly. So a reverse zoonosis would be if the virus originated in the human, and then that virus that originated in the human then is able to be transmitted to a different species, to a different animal. So if it went backwards to the bird, or it went backwards to a bat or a pig or something like that. So we have zoonoses, and then we also have the reverse zoonoses. So let's talk some more about the structure of a virus. So first of all, when we are talking about virions, or a viral particle, they're very, very small. So that's obvious because we talked about how it takes an electron microscope to see it, and it goes through filters that actually catch bacteria. So if we talk about virions, remember these are viral particles. So they're about 20 to 900 nanometers in size, so quite small. What they consist of is a nucleic acid. So we already mentioned nucleic acid. Remember, they have either RNA or DNA, never both. And then surrounding that, they have that capsid. So this is our protein coat around the nucleic acid. So they don't have anything like a cytosol. They don't have any organelles of any sort. What they have is this nucleic acid, and it has the capsid around the outside. So now what could be included with this nucleic acid at times, so they have either the RNA or the DNA, but it may also include some enzymes, very few enzymes though, so maybe like 1 or 2 enzymes that are going to allow it to hijack the machinery, essentially. So we have this nucleic acid, maybe some enzymes with the nucleic acid, and then on the outside of that we have the capsid. And the capsid is made up of these proteins, and these proteins are called capsomeres. So these are the proteins that make up that protein coat. And so it's like little tiny proteins that are all going to come together to make up kind of this shape around the RNA or the DNA. So this, of course, would be three-dimensional. So we have all of these individual proteins that are going to be around the RNA or DNA in all 3 dimensions here. And that's going to be our individual, these are individual capsomeres. And then the capsid is the entire outer shell made out of proteins. And then in the inside here, this is what I am drawing as our nucleic acid, the RNA or the DNA, and perhaps an enzyme or two to allow the virus to infect the next cell or to hijack the machinery. Now, if we have just the nucleic acid, maybe an enzyme or two, and then the capsid, this is what's called a naked virus, or also called a non-enveloped virus. So this is, again, only the nucleic acid and the capsid. So if it's the nucleic acid and the capsid, it's called a naked or a non-enveloped virus. So then on the other hand, we have our enveloped virus. So an enveloped virus has the nucleic acid, has the capsid, and it's surrounded by that bilayer. So surrounded by a lipid bilayer, so like a plasma membrane. And so the viral envelope is what that piece is called. That's why it's called an enveloped virus. It's enveloped by this lipid bilayer. And then that lipid bilayer is called the viral envelope. So our viral envelope is just a small portion of the phospholipid membrane that it got from budding from the host cell. So when we make, when the virus makes lots and lots of copies of itself utilizing the machinery inside of a cell, and then it bursts out from the cell, if it bursts out in such a way to where it grabs a small portion or piece of the plasma membrane on its way out, then it becomes an envelope. So it's an enveloped virus. If it doesn't, then it would be a naked or a non-enveloped virus. So a small portion of the phospholipid membrane obtained when virion budded from the host cell. So then with both of these things, both the naked virus and the enveloped virus, we can also have what are called spikes. And these spikes are made up of proteins, made up of glycoproteins, and they're going to extend out from either the capsid, if it's a naked virus, or from the viral envelope, if it's an enveloped virus. So these are protein structures, namely glycoproteins. And what they do is they allow the virus to attach to a cell and then allows it to enter the cell. So it's going to hook up with some of the receptors on the outside of a particular cell, and then it's going to then allow itself to get inside of that cell and infect that cell because it has these spikes on the outside of it. So they often have receptor sites on the end of the spikes so that they can attach to the cell and bring themselves in. So the spikes are protein structures made up of glycoproteins that allow the virus to attach and enter the cell. All right. So if we took kind of my drawing up here and if I drew it down here again. So let's say we have our RNA or DNA here. Then we have our capsid surrounding that. So remember, our capsid is made up of all of these different proteins called capsomeres. So we have these proteins that kind of create this shell around the RNA or the DNA. And then this could be an example of a naked virus. However, if we then added the glycoproteins embedded in the capsid, then this would be a naked virus that has spikes. And so remember, then we would have a receptor site on the end here that would allow it to attach to a cell and infect it. So now if we have an enveloped virus, then we would still have the RNA or the DNA. We would still have our capsomeres here, making up our capsid. Of course, all of this is three-dimensional, right? So it would be kind of, in these drawings here, we would have a ball, because these would continue three-dimensionally. And then recall that at this point we wouldn't have the spikes because we would have our plasma membrane. So we would have our bilayer on the outside of this. So we'll say that kind of this is our bilayer here, our plasma membrane that the virion particles grabbed on their way when they were getting out of the cell, when they were budding out from the infected host cell. Then we could have our glycoproteins embedded in that plasma membrane. And so then this would be an enveloped virus that has spikes. And not all enveloped viruses have spikes. Not all naked viruses have spikes. Some have spikes and some don't have spikes. So it really just depends on the way that the virus itself infects the cells. Now with these drawings, I kind of drew them round, so they would end up kind of being a ball shape. But that's not true for all viruses. So there's kind of three general categories for the various shapes of the virus. And the shape of the virus is based on the shape of their capsid. So the first one is helical. And when we have a helical shaped capsid, this is going to be a rod shape. Actually, I'll just go down here. So our helical is rod shaped or cylindrical. And so the capsid itself is rod shaped or cylindrical, and then it has the genome inside. So it's normally just inside the length of the capsid. And it actually kind of, the structure, the way it's structured is kind of like an old school telephone cord, or as its name implies, helical. So it's going to be our round shaped here, but then it's going to go, in the different planes here. So we actually end up having something that looks like this, kind of a long tube shaped helical virus. So all of this then is going to be made up of capsomeres around and around and around and around, all of these individual proteins, and then inside of the tube is where you're going to see the RNA or the DNA. So these are helical. The second type, so one is helical. The second type is called polyhedral. And with polyhedral, then this is a many-sided virus. And oftentimes this could be icosahedral or an icosahedron. And an icosahedron is 20-sided. So it's kind of like a 20-sided die, if you've ever seen that, or something that's similar to a soccer ball. So again, three-dimensional, and that's kind of what I had drawn in the earlier ones, but not with the angles there. So if you can imagine, I'm not going to be able to draw 20 sides, but something like a soccer ball like this, but then in 3 dimensions. And these would all be our capsomeres. And then we would have our RNA or DNA in the inside. So that would be our polyhedral. So oftentimes it's a icosahedral, which is 3-dimensional or 20-sided structure, something like a soccer ball. Our third type is what's called complex. And complex basically just means anything that doesn't fit very nicely in helical or polyhedral. And so in our complex, oftentimes what we'll see is we'll see a polyhedral head. And then when we're talking about this example is our bacteriophage here. So bacteriophage, this is the structure specifically of a bacteriophage. We have a polyhedral head. Then what we have is this sheath that comes down from the polyhedral head, and then it includes tail fibers and tail pins. And the RNA or the DNA is in the polyhedral head. So it ends up looking something like, again, my polyhedral, boop, like that. We have our DNA or RNA in here, and then what we have here is the sheath, which is this piece here that comes down from the polyhedral head, and then it's going to have different tail fibers on it. Kind of make it look like this. So this is a complex shape. This is a bacteriophage here, what a general bacteriophage looks like. So we have the different kind of shapes there. It's not just one giant thing. We have the polyhedral piece. We have the sheath, which is like the helical piece. And then we even have those tail fibers there as well. So lastly, let's just talk about the classification and taxonomy of viruses and of viral diseases. So classification and taxonomy of viruses. So there is a group of people called the International Committee on Taxonomy of Viruses. So the International Committee on Taxonomy of Viruses, otherwise known as the ICTV. So the ICTV is the group of people that are coming up with these classifications, coming up with a way to classify and place these viruses accordingly. So the reason that this is so difficult is because viruses tend to mutate very, very quickly. So even within a patient or within a person that's infected, the virus can mutate time and time again, let alone between one host and the other host. So it can be very difficult to go by just the RNA alone or just the DNA alone. So these people here, the ICTV, have gone through and they are classifying, viruses. And so when they're classifying, they are using things like genetics. So they are using the RNA or DNA, but it's a little bit more difficult because sometimes we have that mutation. They're also using chemistry, so crystallization of the various viruses to take a look at them and see what's different. They use morphology. So morphology meaning the way that it looks, right? So the shape of it, so helical versus polyhedral versus complex, and also the mechanism of multiplication. So how is it that it goes about doing what it does? How does it multiply itself? So what are the mechanisms associated with that? What kind of enzymes does it use? How does it hijack the systems? How does it multiply itself? So they're using all of these different things to kind of come up with a classification for viruses. And so far what they've come up with is that we have an order, family, genera, and then sometimes species. So similar to our binomial nomenclature that we spoke about earlier in how we identify and we name things, but even going further up, all the way back up to our domains, when we're talking about eukaryotic and prokaryotic organisms, recall that we have, domains and kingdoms and order and family and species and genera, all of these different classifications in order to narrow things down so that we can make sure that we're all on the same page. We're all talking about the exact same thing at the exact same time. We have the same here with viruses, but it doesn't go all the way up to domains like we see with the other things. Because remember, when we're talking about domains, we're talking about living things. And remember, a virus is not a living thing, so it doesn't have a domain. It doesn't fit into any sort of kingdom in that way. It's its own separate thing. So our classification for viruses includes order, family, genera, and then sometimes species. When we are writing these things, all are italicized. So we italicize all of them. Remember when we did bacteria, we italicized that as well. When we are taking a look at the family and the genera, what we see is that in family, I'm going to go through here, it's going to end in viridae. So, for example, the example the book gives is Parvoviridae. So that is the family. And when we get to the genera, it ends in virus. So in the Parvoviridae family, we have a virus called the parvovirus. So when we're talking about the genera, it's going to end in virus. When we talk about the family, it ends in viridae. And then sometimes we go further down to species. So the example that's given in the text is Pandoravirus, so that's the genus, Pandoravirus dulcis. So the dulcis would be the species, and then we know that Pandoravirus is the genera or the genus because it has virus at the end. So this is how the ICTV has classified viruses. Now there is an alternative to the ICTV's classification. That's called the Baltimore classification. And the Baltimore classification is also based on genomes, and it's based on the mode of replication. So how it replicates, how it makes copies of its genetic information. So this is just another way to classify things. So they are often kind of grouped by characteristic, however. So when we talk about viruses, when we don't generally just kind of go by the ICTV type of taxonomy or the Baltimore classification. When we talk about viruses, we tend to group them by characteristics. So grouped by characteristics. And that doesn't mean that we don't use those names. We do. We use the names that the ICTV put out. But when we talk about viruses, we kind of group them based on these different characteristics. So first, whether they're naked versus enveloped. Also, whether or not it has a single strand or double strand of nucleic acid. Whether or not that particular nucleic acid is, again, DNA or RNA. Whether it is segmented or non-segmented. So when we talk about segmented versus non-segmented, what we're talking about there are the genomes. And then lastly, if it is a positive strand genome or a negative strand. And we'll talk more about what it means to be a positive strand or a negative strand in the next chapter, or the next section. So, for example, an example that's given in the book, so the herpes virus, for example, is classified as a double-stranded DNA enveloped virus. So, double-stranded DNA enveloped virus. So when somebody says, oh, the herpes virus, what comes to mind then? Because it's often grouped in this way. When a person is thinking about that herpes virus, they're going to be thinking about the fact that it's a double-stranded DNA enveloped virus. So what that means, and we're going to get more into what that means. We already know that enveloped means that it has the envelope. We know that it has DNA and it's double-stranded. And we know that by indicating that this is a double-stranded DNA enveloped virus, we actually now know, or will know when we go over that, how it's going to get into the cell, how it's going to make copies of itself with the kind of processes that it has to go through, and then how it gets out of the cell. So another example, HIV. So HIV is a positive single-stranded RNA enveloped virus. So in this case, since it's a single strand, we say it's a positive strand or a negative strand. In this case of HIV, it's a positive single strand RNA enveloped virus. So if it's a positive strand, it has the positive in front of it. If it's a negative strand, it has the negative in front of it. If it's a single strand, we see the small, SS. If it's a double strand, it has DS in front of it. And then, of course, DNA or RNA. And then lastly, we can talk about how we classify viral diseases. So classify viral diseases. So we just talked about how we classify viruses, so based on their genome, et cetera, et cetera. Then just quickly about classification of viral diseases. So the ICTV, again, they have classified the viruses. They have worked with the International Classification of Diseases, or the ICD, which is a part of the WHO, the World Health Organization, to classify the actual diseases that these cause. So let me write those down. So the International Classification of Diseases, which is the ICD. And the ICD is maintained and is updated by the WHO. So maintained, updated by the World Health Organization, or the WHO. So the World Health Organization, which is, as its name implies, an organization that has people from all over the entire world in it. They come together and they talk about the health of the world, how things are moving from, one country to another country, etc. And they are in charge of this International Classification of Diseases that was kind of started by or has input by the ICTV. So the ICTV, of course, is classifying the viruses themselves. So they're working with the WHO to come up with this International Classification of Diseases, or the ICD. And what this is an alphanumeric code. Which is about 6 digits. And then it is assigned to the disease. So alphanumeric has alphabet and numbers, and that code is about 6 digits, and then they assign that to a particular disease. And the importance of the ICD is because this is what's used kind of all over the place. So as soon as a person comes into a doctor's office, if the doctor needs to order labs, then they'll order the labs based on the virus infection that they think that it is. So then they would use this ICD. So when they order the labs, the ICD number or alphanumeric code is going to be written on there to say, like, this is what we're looking for, this is what we think it is, so the ICD code is on there. So then at that point they can see the ICD code, so for the laboratory tests, then also for the medical laboratories, so that they can identify what tests need to be done for that particular ICD code. So to send to labs or at the lab. And then that person, so then after they would do those labs, say they confirm that that is it, so then that ICD code is going to be used in the healthcare system. So when the doctor then gets those lab results back and they talk to the patient, they can write down that that is in fact the code, that that is the correct disease. They use the ICD code. Then that ICD code is going to be used by various medical coders, when they are assigning things, when they're talking about the procedures that were performed. This is used by medical billers or insurance companies, so that the particular labs that were done are going to be matched up with the ICD. It's also used on death certificates, so that if it was due to a particular viral infection, then that ICD would be a part of that. And then lastly, epidemiologists. And epidemiologists are those that are going to be studying disease and how it spreads. So they're the ones that are going to be taking a look on, whether or not it's an endemic or a pandemic, and studying the way that these diseases are moving throughout, the state or the city or the country or the world. And so then they're going to be using the specific ICD code in order to watch these things and classify them. And then it also is used to calculate things like morbidity and mortality and things like that.